we're going to move over, in which case, to hear from Ben Jones, who's going to talk about sourcing rare frozen red cells. I hope it's still okay for everyone to stay with us. We've got plenty of time. Ben's an experienced biomedical scientist with a passion for investigating the most complex red cell antibody investigations in the world. Loves using a wide variety of serological and molecular techniques, and enjoys training new members of the IBGRL team and seeing people's passions in the field grow. So thanks very much to Ben for this final talk of the day, and we've got time for questions afterwards.
Everyone see that okay?
Absolutely fine.
Brilliant, wonderful. So good afternoon, my name is Ben Jones. I'm presenting on behalf of the NFBB today. I will say that I do not work at the NFBB, but they are under some clinical requirements that requires their time to be elsewhere, so I'm standing in. So apologies if I'm a little bit working on my feet today. So the National Frozen Blood Bank aim is to provide extended storage of rare phenotype red cell donations. They are the only frozen blood bank in the UK and are based in Liverpool. NFBB serve hospitals throughout the UK and are occasionally international referrers as well, providing thawed and washed rare red cells. A very small department which offer a 24/7 on-call service, so can deliver thaw and wash red cells day and night, 365 days a year, most of the time. Currently under management by the International Blood Group Reference Laboratory, which is where I work. So it all starts with a donor who takes time out of the day to kindly donate blood. The donations are sent to the nearest manufacturing site. There are 4 of these sites across the UK where these units are manufactured into various different components, such as red cells, platelets, and plasma. But NFBB are only interested in the red cells, though, particularly the rare ones. The rare units appear on a daily list, which NFBB check and order to freeze if needed and hold it until it is required by a patient in hospital, for example, the McLeod case that we just saw. And they can keep frozen units in glycerol for up to 30 years. NFBB rely on rare donors whose cells lack clinically significant antigens, which are present on nearly all other donors in the population. So rare donors make up 0.1% of or less of the UK population. They are identified as rare when the samples are put through the standard testing departments within NHSBT, where this is by identifying an atypical antibody in that donor, or a rare antigen, usually lacking type, is detected and confirmed by the International Blood Reference Laboratory. Donations from some ethnic origins, such as Black and Asian backgrounds, and all RO donations are selected for further phenotyping and rare antigen screening. Once a donor has been confirmed as having a rare type, their electronic record is flagged with a VIP or RAR code. This ensures that any future donations appear on the daily list and are frozen by NFBB. The current stocks of rare units in NFBB are approximately 1,000. This comes from a small pool of 530 rare UK donors. Red colour on this chart indicates low stocks or no stocks, blue is medium, white is okay target-wise, and green is stocks where there has been an issue such as 24-hour expiry or includes an imported unit from another country. So you can see from this list, there's lots of different things that are stocked at NFBB, and we don't have abundance of quite a few of these types, and it can be affected by the ABO group. On this list, it's actually only the group O rare types. Now I'm going to focus on 3 rare types that we will aim to freeze every donation of at NFBB. First one is the dash-to-dash. It's a variant of the RH system where big C, little c, big E, and little e antigens are all completely lacking. And we have 1 UK donor who is a group A, and there are only 110 donors worldwide, with around 80% originating from Japan. It's an extremely rare type, and it can be given to patients with very specific RH-related antibodies, for example, anti-RH17. Next is the Bombay blood group, which I'm sure many have heard of, AKO-H. Cells from this type lack the H antigen, which is the building block for the antigens of the ABO blood group system. Therefore, patients with the Bombay blood group type who are in need of transfusion can only receive from this type, so they can only receive other Bombay units. This is why it's important for NFB to collect and freeze every Bombay donation counter, which is quite challenging as there are only 3 donors in the UK. Currently have only 4 units in stock in the frozen blood bank, as seen in the image, which are from 2 of the donors. We have forward and washed 39 units for patients since the start of the frozen blood bank of this type. Finally, we have the IMB negative blood donation type. There are only 11 UK donors for this. We freeze every IMB donation no matter the type. We have forward and wash 14 units for patients of this type since the start of the frozen bank. Thing to mention regarding all three of the types that I've just mentioned is that when we freeze them, we give them an ultra rare status on our database, in which, if issued to a hospital, they are tagged up with a special label so that if they are not used, they can be returned to us. They can be used for research purposes by IBGRL as well, or by the reagents department. That was the rare types that we freeze, but how is this achieved? You cannot just freeze red cells without a cryoprotectant. Therefore, we use glycerol. Glycerol is essential to prevent dehydration and mechanical trauma to the cells during freezing. Without it, the water inside the cells, when frozen, forms sharp ice crystals, which could then puncture the membrane. Glycerol is membrane permeable, therefore it enters the cells as water leaves during freezing. Equilibrium allows cells to regain original volume, and there is also a low proportion of electrolytes, so the cells are not overexposed to solutes and there is less ice formation. It was only really realized that in the 70s that glycerol could be used for more for long-term storage of frozen red cells. It offered storage solutions for unexpected high blood demands during operations or disasters, rare donor blood requirement, or if we needed to store autologous blood. So the process of adding glycerol to the red cells is called glycerization. This enables them to be stored for up to 30 years. It's an automated closed system process. We glycerize during normal work, well, NFBB glycerize during normal working hours, Monday to Friday, 9 to 5. There is a 4-hour window where it has to be completed from the time it's removed from cold storage to the time it's placed in the freezer. And units need to be less than 5 days old or less. And this is to ensure that O2 transport function of the red blood cells is maintained and is at its peak when frozen down. So here's a really wonderful piece of equipment that I've become very familiar with in the last 24 hours. This is the ACP215 Immunetics Automated Cell Processor. It works by calculating the amount of glycerol to be added. So it literally just does the work, does the hard bit for us. So you input the hematocrit and the weight of the unit, and it uses disposable kits, so nothing is reused to maintain sterility and all connected using sterile wielding devices. The unit is put on the machine for about 10 minutes. As you can see in the image, the glycerol was spiked with one end of the kit and hung upside down to allow flow of glycerol. Red cells are attached at the other end of the kit and placed on the shaker. In between the two is a pressure monitor, a bacterial filter, and a pump to control the flow of glycerol to allow it to be introduced at the correct rate. In the 4-hour glycerization window, the units go through 2 spin cycles in the century. The first spin cycle is before it gets put onto this ACP machine, and this is to remove the SAGAM that would have been added during the manufacturing stage. And the second spin is after it has been on the ACP to remove the excess glycerol. A HCT is also obtained after each spin cycle. The first is so that the correct HCT can be added into the ACP machine, and then the correct glycerol can be added in to the unit. And the second is for when after the glycerol has been added, so that when we come to deglycerization and thawing of units, they can be put on the same machine so that the glycerol can be removed safely and ensure that the red cells remain intact. And the SAGAM and the excess glycerol is removed after the centrifugation process using a manual press, which you can see on the right image here, which literally you spin it down and then it pushes the supernatant off at the top. Once the unit has been glycerized and labeled, it's vacuum packed and then boxed into a cardboard sleeve. It's frozen flat for 24 hours before in a signed position. Here is how they are stored in these freezers, and they're stored upright and at -18 degrees. This is where they stay until they're needed. Each row contains about 17 racks, and each rack contains 4 units. There's 8 freezers in total at NFBB, and the oldest units we have in storage, or I believe were had in storage, was the Kelmel units, which were 22 years old. So we've got the units frozen. So now we need, obviously they're no use unless we can thaw them and provide them to patients when required. This process is called deglycerization. So frozen red cells to remove the glycerol so that they can be issued to patients. So we have 2 ACP machines which carry out the process of doing this. It takes about 4 hours for the whole process from start to finish, the 4 units. So you can do 2 units within 4 hours at NHSBT. The first step before we start to thaw and wash any units is to receive the order from the RCI at NHSBT, and this order has to be authorized by an NHSBT consultant. Units are located from a database and found in the freezers, thawed to 35 degrees in a plasma thaw, and then attached to the ACP machine using the deglycerization kit. So it's the same machine for using the deglycerization. Here's the ACP again. The parameters that need to be added are the weight and the thaw pack and the HCT obtained after the glycerization process. Deglycerization works by using a range of osmotic tolerance to remove the glycerol safely. So the intracellular and extracellular concentrations must revert as equally as possible to avoid hemolysis. So firstly, a 12% NaCl solution is added, and this is hypertonic, so the cells initially swell and then quickly shrink as the glycerol moves out of the cells and the equilibrium is reached. Secondly, a 0.9% NaCl is added, and this is isotonic, so the solution removes the remaining glycerol and washes it away from the solution. Then after it's all been removed, sargem is added back into the red cells to preserve them, and it approximately takes about 1 hour on the machine to complete all of the wash cycles. So our rare units go through a lot. So just to summarize, from starting their journey as whole blood, being processed as red cells, selected by us at NFB, glycerized, stored at -80, thawed, and then needed by a patient in need of transfusion. So being the only frozen bank in the UK does pose some challenges. We do encounter them. Some rare types only have 1 donor, some don't have any in the UK at all. Our only McLeod donor is now 77 years old. She's actually probably older than that now, from when this presentation was last given. Another challenge for us is having to freeze within 5 days of the units being bled. It becomes a medical concession, but we do freeze these units still because some of these units are extremely rare and they need to be frozen down for patients that are really in dire need of them in the future. So they can be frozen under concession and then released under concession, but for those patients, sometimes there's no other choice, so that is by far the best option. But for that to happen, the HB does need to be greater than 30 grams per unit. So very quickly, I'll just show you a quick clinical case study, a little bit different than the McLeod one. So this was a sickle cell disease patient with a complex blood requirement. They had antibodies to big C, big E, big K, or they had been identified previously anyway, and little s and JSA. So it was first seen in NFB in 2010, and there had been blood imported from the USA. So they received a total of 47 units, which is quite a lot, and some of these had been brought in to NFBB and frozen down for when this patient required them. So there was a major collaboration between IBGRL and the medical team in Birmingham, where this patient was. Donor call-ups were actually issued so that we could get more donors to get frozen down at NFBB for this patient. And this highlighted some of our U-negative donors that we had in stock that we could actually bring in to provide blood for this patient. And thankfully, the patient recovered and has not needed transfusion since 2020. The second case study is, I almost remember this one. A patient in her 90s suffering anemia needed an urgent transfusion. Not only did she need Kel negative blood, she was also in a hospital in a remote UK island. So we had 3 units, all from 1 donor, and they were the oldest units in the bank, bled in 1999 and 2 bled in 2000. Usually the thaw and wash shells have an expiry date of 72 hours, but as these were so old, the expiry date had actually been reduced to 24 hours. So it's not a very large time frame to get those units to the patient. So collaboration was needed between us and tissue service, as the unit needed spiking in a clean room, usually with a sterile connect the donation pack with a transfer pack in our lab, but this couldn't be achieved as the line was really short. Collaboration was also made between us and the IT experts to get the unit labeled correctly and issued in time for the flight to Scotland. So quality assurance and medical staff were also on hand to approve the issue of the unit to support procedure. IBGRL were always providing expert advice regarding the typing of the unit. And bearing in mind there are only 3 units in the whole of the UK, the first one was unfortunately leaked as it was being thawed, but fortunately the second one was thawed and washed successfully. All was not lost though, as the leaked unit was sent to IBGRL to help save lives by enabling storage to find other donors around the world. The transfusion was successful and the patient was discharged a few days later. A surprising twist in this story is that the patient was actually the donor of the units. So after donating them almost 20 years previously, she had received one of them back when she needed it the most, which I think is nice. So that is my little talk on NFBB, and thank you for listening.